cancer cells are notorious for their ability to divide uncontrollably and generate hordes of new tumor cells. Most of the fuel consumed by these rapidly proliferating cells is glucose, a type of sugar. Scientists had believed that most of the cell mass that makes up new cells, including cancer cells, comes from that glucose. However, MIT biologists have now found, to their surprise, that the largest source for new cell material is amino acids, which cells consume in much smaller quantities. The findings offer a new way to look at cancer cell metabolism, a field of research that scientists hope will yield new drugs that cut off cancer cells' ability to grow and divide. If you want to successfully target cancer metabolism, you need to understand something about how different pathways are being used to actually make mass, says Matthew van der Heijden, the Eisen and Chang Career Development Associate Professor of Biology and a member of MIT's Koch Institute for Integrative Cancer Research. Van der Heijden is the senior author of the study, which appears in the journal Developmental Cell on March 7. The paper's lead author is MIT graduate student Aaron Hozios. Since the 1920s, scientists have known that cancer cells generate energy differently than normal cells, a phenomenon dubbed the Warburg effect after its discoverer, German biochemist Otto Warburg. Human cells normally use glucose as an energy source, breaking it down through a series of complex chemical reactions that requires oxygen. Warburg discovered that tumor cells switch to a less efficient metabolic strategy known as fermentation, which does not require oxygen and produces much less energy. More recently, scientists have theorized that cancer cells use this alternative pathway to create building blocks for new cells. However, one strike against this hypothesis is that much of the glucose is converted into lactate, a waste product that is not useful to cells. Furthermore, there has been very little research on exactly what goes into the composition of new cancer cells or any kind of rapidly dividing mammalian cells. Because mammals eat such a diversity of foods, it seemed like an unanswered question about which foods contribute to what parts of mass, van der Heijden says. To determine where cells, including those in tumors, were getting the building blocks they needed, the researchers grew several different types of cancer cells and normal cells in culture dishes. They fed the cells different nutrients labeled with variant forms of carbon and nitrogen, allowing them to track where the original molecules ended up. They also weighed the cells before and after they divided, enabling them to calculate the percentage of cell mass contributed by each of the available nutrients.